Alright, so when I say phylogeny and phylogenetics, what do I mean? So phylogeny really means the evolutionary relationships among groups of organisms. Now that sounds fairly straightforward, but determining the evolutionary relationships between organisms isn't always as clear as it seems. So if you think about the concept of the tree of life, a tree is a useful way to consider evolution, not because a tree evolves, but because a tree has a structure that conceptually is a good way of visualising perhaps how organisms might be related. So if we have a tree, we have the tips of the branches are the present. So now, if you like, and there'll be a number of organisms at the tips of those branches that are related through the way the branches are connected back down the tree, which means back through time. Phylogenetics then is the study of the evolutionary relationships amongst groups of organisms. So there are different ways of doing that. These ideas are not new, and it's, I guess, around the sort of time of the birth of what we might call modern biology, all of these ideas were being considered. So perhaps in around the 1600s is when modern biology, as we like to call it, the study and classification of animals and looking at their relationships really began. And it began as a way of trying to make some order of life. People had recognised for a very long time that there were different types of trees, for example, and some look similar and some look different. Then questions arise about, well, are trees related to things like grasses and other green things and so on? Right, so these questions have been around, but in terms of a formal structure to think about and compare organisms, it really began when taxonomy as a modern science began in the 1500s and 1600s. So a way of ordering information. So here's a tree from fairly early on, the pedigree of man proposed by a guy called Haeckel. It's somewhat stylistically a tree, but if you actually work your way up that tree, even though there are many minor details that are probably misleading, the general order of things even was pretty well understood back then. We may not have had lots of evidence to support it, but there were ways of looking at organisms and comparing them and determining which ones were related and therefore probably evolved from common ancestors. In the 1850s, Darwin, one of the sort of fathers of evolutionary thought, of course, also sort of these things. There's a little quote there where we talk about a time when we shall have fairly true genealogical trees of each great kingdom of nature. So genealogical really is a word that means phylogenetic. So hopefully genealogically reflecting the true evolutionary history as opposed to something we might interpret from looking at something else, like the structure of organisms, for example. So if we want to come back away from evolution for a minute and think about ways in which we can approach understanding the relationships and, by proxy, the evolutionary patterns that might have arisen or led to current life, one way of structuring it is what you might call character and taxon analysis. So this basic concept and a way of organising information about organisms, or in fact any object, has been around for a long time, and it doesn't just apply to evolution. It's really a mathematical approach. So if we think about things we can measure, we can call them characters. So if we come back to people in the classroom, for example, we can talk about blonde hair, black hair, blue eyes, brown eyes. They are characters. Alright? Taxa, or taxon, tax is plural, taxon is a single object. They are basically, in biology, any species, or in fact any individual for which we can record a series of characters. So characters are information about the same feature. Taxa is the object which we might be describing and recording information from. So in most phylogenetic studies, our taxa are the same as biological taxa, species, genera, things like that. Characters will be something we measure. So it could be leaf shape, for example, if we were doing it with trees. The ones on the left here are different to the one behind us there, for example. So we need to find some way of measuring that information. So we can use any type of data in a character analysis. Right? So we can record them and arrange them as a series of characters that compare two or more taxa or two or more objects. The main thing about recording characters is to try and record the same feature. So for instance, if we were to record, say, the number of points on the ends of the leaves as a character associated with these trees versus that, we'd need to be fairly sure you're comparing two things that are the same measure. There's no point trying to record, or it's terribly confusing and doesn't work terribly well if, for instance, if we were to record hair colour and eye colour, right, amongst the individuals here in no systematic fashion. So let's say, well, now we've got blue eyes on this side of the room, but over here we've got brown hair. So to compare eye and hair colour, you're comparing apples and oranges. All right. So characters need to reflect the same basic information about the individual we're measuring. So that sounds pretty obvious, but it becomes less obvious when you're talking about characters we're less familiar with. Alright, so if we measure a series of the same measure from different individuals or different taxa, they can either be fixed, that is, it's the same across every individual that we measure, or it can be variable, which means, again, eye colour, there's different categories of eye colour in this classroom we can measure and record. So we continue to build up a set of characters, and we could measure 100 different things for the visual appearance of every individual in this class. We would have 100 characters across 9 individuals. So what type of data can we use? Well, I've given a few examples of morphological data, but you can use anything. Anything you can measure can be used in this form of analysis, and in theory can be used to look at the relationships and phylogeny between organisms. Right? So hopefully if you have a set of identical twins in the classroom, and they truly are close to identical, and you measured thousands of different aspects of that person's appearance, you would find that they would be very similar. They're very rarely completely identical. There are minor differences. So therefore they would share a large number of the same character measures compared to two different individuals in the classroom. But we can also use biochemical data. So it could be, say, the level of expression of a particular gene or a protein in their cells. It could be molecular, so it may be DNA, RNA, protein sequences. Right. Anything that varies between individuals can tell us something about the relationship between them. Now, if the character we measure isn't variable, an example I think I had in the last slide was the number of legs, assuming we all haven't lost one or have a birth deformity, we're not going to have a lot of variation in most populations, right, if we say how many legs. In fact, even if or how many limbs, we could go right across quite a large part of the mammalian group, and we still wouldn't find much variation, because pretty much most mammals are built on the same body plan. They've got 4 limbs. So that wouldn't be a choice of a character that's going to be very useful within the mammals, for example, because we can't put them into different categories. So those characters have to contain information, ideally, and for studying evolution, those characters must also contain some information that is correlated with the evolution of the group. All right? So if the group of organisms really does share common ancestry from an evolutionary perspective. They will share some features, structural, morphological, biochemical. Others will have differed over time. But if we measure things that actually are not strongly correlated to the relationships and the evolution of the organisms, then clearly it will just be measures of similarity that are derived from something else. So a good example might be convergent evolution. You might say something like a flying possum versus a flying marsupial possum. Actually quite distantly related in the mammals, but if we were to measure basic morphology, they actually look quite similar. Right? So there we might be misled by the fact we might measure morphological characters that are actually not directly related to their evolution from a common ancestor, but are actually related to their adopting a similar lifestyle, for example. So what we want are homologous characters. So homologous means we're measuring traits or characters about a taxon that are the same. Again, it seems intuitively obvious for things we're familiar with. If we're going to count the number of limbs, we can mostly identify which are limbs and which are not. But depending on which group of organism you're looking at, it doesn't necessarily become that clear that you're comparing the same thing. Right? So we want to compare the same thing, and in terms of phylogenetics, we're talking about comparing characters that are homologous. So homology is a similarity that arises from inheritance. Alright, so a good example might be primate skeletons, which are homologous. We share a certain body plan in the way our skeletons are structured that is a result of our ancestry from a common ancestor that formed the primates. So some years ago I was wandering around in a museum in London, and there was a display in there of an ape skeleton and a human skeleton. And you look at those and there's a hell of a lot of similarities in the way those skeletons are constructed. So we could use skeletal homology and estimate how similar apes are to humans, or monkeys are to apes and humans, by measuring different aspects of the skeleton. So the skeleton has thousands and thousands of things we could measure about it that vary. So for instance, you could look at pelvis length and ratios, which would show you that humans are somewhat different to apes. There are some areas which will look very similar, like clavicle bones and so on. And in fact, there's a whole science, evolutionary science, that actually does exactly this on skeletons, because it's one way of identifying the relationships between fossilised bones that are dug up. It's how they work out whether what's being dug up is something new or belongs to one of the existing described humanoids or apes. Right, so that's an example where the homologies are relatively clear. All right, but if we go a little more broadly outside the primates, this is basically the same limb in a human, a cat, a whale and a bat. It's been drawn and coloured to show you the homologous bones. So the bones that are actually the same bone in the highly modified structure of the limbs. Alright, so here we've got the forelimb, the arm, that's the front leg of a cat, that's the front leg of a whale and a bat. Highly modified, but you can see the same basic structure. You've got fingers in each case, but in some cases the shape, length and level of fusion of the bones is quite different, right, which gives the whole limb a very different look and function. Right, so if we were to compare homologous characters, if we were doing skeletal analysis on these four organisms, we would need to be comparing the same bones with each other. No point comparing, say, the length of this bone with the length of one of those bones if it's from a different phalange, different finger, because it's not the same structure, for example. All right? Or we wouldn't be trying to compare this bone with one of those in there because it's not the same structural element. So homology is really important. Again, reasonably obvious when you can identify which character is actually the same structural element, but not so easy when we get to something like DNA and RNA. All you've really got to go on is A's, G's, C's and T's. All right? So another quote from Darwin, basically suggesting that characters that naturalists consider as showing true affinity, i.e. they're homologous, are those inherited from common parent. Alright, so all true classification, which we could call classification and taxonomy, in reality should be based upon homologous characters, effectively the phylogeny. So how things have evolved is the appropriate way to look at the relationships among organisms. So that's the goal. The reality is, in some groups we're close to that goal. The way taxonomy and classification is structured in some organisms is actually probably pretty close to what the true phylogenetic history is of the group. In other groups, it's probably a long way off, and particularly, certainly in fields I work, so it's microbial eukaryotes and algae and so on, there's a whole lot that's wrong with the taxonomy because there's actually quite a bit of difficulty determining what are homologous structures and what are not. Right, so if we move on a little bit from very early thoughts about evolutionary analysis to a guy called Linus Pauling, who tended to be a bit of a polymath. He contributed a lot to lots of different fields, including early molecular biology. This paper basically foreshadows the use of molecules as a record of evolutionary history. The basic philosophy being that something was, two organisms were relatively closely related, they would share much more information in the way the molecules that make up the cell are constructed. And they gave the examples in that paper of the macromolecules that were probably the best bet in terms of retaining genetic information and therefore information about our evolutionary history. DNA, RNA and protein, and that's exactly where we are now. That we're using these long polymer molecules because they carry information about the evolutionary history. Alright, so that's really an introduction to the basic concepts. What does this mean when we get to our DNA sequences this afternoon? Well, we can think about sequences in a phylogenetic analysis in the same way. It's a series of taxa and a series of characters. So a few weeks ago we had a play around with a little bit of data in the program that we're planning to use. It's possible to arrange 6 individuals from which we each have a DNA sequence for the same gene, because we want to compare the same gene, because we want to compare homologous characters. We can't compare one gene with another because they don't share an evolutionary history. It's possible they may do. It may be a copy of the gene. It's evolved in the past. Then doing phylogeny gets a little more complicated. You have to consider gene families and things like that. If you think about a single gene, there's one type of that gene on the genome of the individuals. We sequence it for 6 different individuals, and we might get this sequence back for each of the individuals. All right. One thing we need to do is work out which characters are homologous or not. We might have homologous genes, right, the same gene, but each position then represents a character. So an individual measure of the character of that organism. So if we've got 45 base pairs in our gene sequence, we've got 45 characters that we can compare. But only if we can arrange them in a way that we can be relatively sure that we're comparing like with like. So that means we need to identify homologous regions, or what are called the same sequence motifs, right? Because when we do so, we can see there are clear patterns, right? Sequence motifs, where you can see there's lots of things that haven't changed much, right? No variation much along here, but here, well, that one's different. Those 2 are different from this one, and these 2 are different. Those 3 there share a T. Right. So we can infer, after aligning our data, that, well, nothing's changed here. So we've got 1, 2, 3, 4, 5 characters that are invariable, constant across our 6 individuals. But we've got a couple here that are variable. We've got 1, 2, 3 different bases across our 6 individuals. All right. So we can look at those and say, well, that's one variable character that we can use to group like things together and different things apart. So we can automatically create 3 different groups. One's with a T at that position, one's with C at that position, one's with G. We keep moving along. Still the same. Oh, we've got another position that varies. All right. So we can now create more groups. This one will split our C group into a couple of different groups. Well, actually, no, they'll be the same, sorry. So this one here will separate our groups containing a T at that position into a couple more. Ones that have a T here and ones that have a G. So the more characters we compare that are variable, the more capacity we have it to distinguish the individuals from each other. So if we count up the number of shared and similar characters, DNA alignment positions, in this matrix, some will be really similar and some will be quite different. So instantly we have a measure there that we can make some determination of things that are more similar and things that are less similar. Alright, so we've got fixed characters and variable characters that we can use. Along here, not all DNA sequences are identical lengths. Right? So some, particularly in non-coding regions or things like RNA sequences that we're working with this afternoon, you can have single, 2, 3 or multiple base pair deletions that don't actually seriously affect the function of the molecule. Right? We're inside a coding gene. 1 or 2 base pair deletions are really uncommon because that'll induce a frame shift downstream. So everything will be out of kilter downstream. So we'd expect 3, 6 or 9, so on, deletions inside a coding gene that codes for a protein. But in this example, we've got a few different deletions of different lengths. To keep all this aligned, see it looks actually really similar along here, we didn't put any what I call inserts and deletions or gaps into our alignment to push this along relative to this one. All of this would then be out of alignment along here. All right. So this afternoon when we do our DNA alignment, it's a process basically of working out where to insert the gaps to keep the bits that are non-variable in the same frame position in our alignment, so that we're still comparing homologous characters along here. So if you set up your alignment with coloured blocks like this for different bases, it becomes really easy to see where things are aligned, and areas where there's a lot of variation or they're poorly aligned become obvious because you can't see those vertical blocks of colour anymore very easily. So if we didn't put these in, everything down this way then is out of alignment again, because there are bases missing in some of these relative to this one. And we can infer from that there's been a deletion in the DNA relative to this sequence here at the bottom, which is the longest sequence. Okay. Alright, so we need to be able to align our data, our DNA or RNA or protein sequences in this manner so that we can determine which are homologous. Alright, so this is homologous, this, even these ones with a difference. We're hopefully reasonably confident at that point we're comparing like with like. It's the same location in that DNA sequence. So alignment's a way to aid identifying which things we need to compare. Alright, so in a DNA alignment, each position in our alignment has 4, in fact 5 possible states. It can have an A, G, T or a C, or it can have a gap. Right? Because we've had to insert one in there to keep everything aligned. That means no base, basically. Right? Proteins are somewhat different because we're then aligning amino acid sequences. There are more than 20 possible states here. Alright, so there's considerably more capacity for information in protein sequence because you've got, instead of 4, you've got 20 different options at every point. And that also creates some difficulty when we come to aligning and comparing protein sequences, because they're naturally more variable. Alright, so how does this process of sequence alignment work? So you'll be able to have a crack at this afternoon. You can just do it by eye. Set up yourself a text file on your screen and just work your way along each sequence, putting in a dash or a space and taking them out and so on to try and make things line up. So for instance, that relatively short set of 6 sequences, I think I actually did that one by eye anyway. Because you've only got a few points that are variable, you've just got to work out exactly where to put the gaps in. And then you'll see this all stays in alignment, this comes into alignment eventually, and then you're trying to work out which requires the most change, or the least change I should say. Right, so we want to insert as few gaps as possible when we do that. Alright, so having colour makes that a lot easier. Trying to do it without coloured blocks is difficult. But when we get to long sequences, or if we've got 20, 30, 50, 100 different sequences to compare and align, that gets really slow and difficult to do because you just physically can't process that information visually. If sequences are really, really similar, so there's not a lot of variation, and the sequences are relatively short, then again, it's actually not too difficult to do it by eye. But as soon as it becomes more complicated, then there are computer automated algorithms that will do this for you. Right, so basically an alignment algorithm, which we'll be running this afternoon and a lot of software will run for you, are mathematical ways of generating what is basically the simplest solution to this process of aligning DNA. So when I say the simplest, the basic principle of most of these algorithms is you want to try and minimise the amount of inserts and deletions that you put in to create the alignment. So it's working in the idea that we want the simplest answer to get to the alignment. And the way it does that is it generates a total score of mismatches. So it goes across the matrix and works out how many changes are required in the solution, which is the alignment. All right? And you can tweak that algorithm by changing the relative weightings. So you can say, I want you to weight gaps much higher than a change between an A and a T, for example, which would force the algorithm to not put gaps in relative to allowing changes between bases. Or we could weight them the other way. Say if there's any variation, put a gap in. Don't allow a G to C transition, for example. All right? So depending on how you set up that alignment algorithm, you can get it to be more likely to put gaps in to assist with alignment, or you can force it to actually not put gaps in. And that's effectively what alignment algorithms do. They just put gaps in the places to bring everything into alignment. Automated algorithms are not always completely reliable, particularly when you get to areas with lots of gaps of different lengths. So we've got sequences that vary quite a bit in length. They're often not great at making decisions about which side of gaps a particular base should sit on. We could make a decision to put this C here in the middle, for example, or over here. And sometimes either of those options are mathematically equivalent in terms of total score or cost to the alignment, but logically it's obvious where it belongs. All right? So it particularly happens when you have a lot of single and short gaps. So you always should take a look across your alignment that's been produced by your algorithm to see whether it actually looks okay. And you can manually edit your alignments if you think the computing algorithm hasn't really done a particularly good job. And you can do that by changing the costs of actions and rerunning it, or you can just change things manually. Having said that, the default alignment parameters that are typically set up in these algorithms are usually okay with most data sets where you don't have regions with lots of variation and lots of inserts and deletions. The molecule we're working with, the ribosomal RNA, does have a couple of regions where there's a lot of length variation between organisms, and there's quite a few inserts and deletions. So there'll be regions where we might want to refine that alignment by eye or by changing the alignment parameters. Alright? So you should always check and adjust by eye if it doesn't look a terribly good solution. Pardon me. So once we've generated that alignment, typically what you want to do is visualise it in some way. The alignment will give you some sense of how similar some things are and how different others are. But it's not a terribly easy way to interpret which things are close to each other or very different, or by how much. So there are a few different ways you can do this. One would be building a phylogenetic tree. So a phylogenetic tree is effectively, mathematically, what's called a network. That's a series of lines that connect points in space, right? Which is what that is that you can see in the background. The important thing about this process is the output, your tree or your visualisation of that alignment, is only as good as the alignment that goes in. So if you have a rubbish alignment, you often get a tree that isn't a true reflection of the relationships or isn't a good reflection of the relationships between the organisms. So you really need to work on, firstly, the data that goes into your alignment, which is data correction, making sure that you haven't got erroneous base calls in your raw data. And once you've got your alignment, the quality of that alignment and how well aligned that data is crucial to the tree that comes out the end. So you've got this stepwise process where you have to pay a lot of attention to each step, because the end point will mislead you if you haven't paid enough attention to the quality of the data that's going in and out of each of these steps. So from our alignment, there are 4 different families of ways we might build a tree-like structure to visualise the relationships. Main one we'll deal with in this unit is what's called phynetic, and that's where we group things based on an overall measure of similarity. So we go back to our alignment, we can count up how many bases are the same and how many are different. We've got, I think there were 45 or 46 bases in our alignment, including the areas where we've got gaps. We can count up, there might be 5 differences between the first and the second organisms, and there might be 10 differences between the bottom one and this middle one, for example. Right? So when we do that, we're generating overall measure of similarity or difference. So it could be 95% similar and 5% different. And we can use that information, either similarity or differences, to determine how similar and how different the organisms are, and use that to say, well, if they're more similar, they must have derived from a common ancestor more recently, because we've had less evolutionary time for those differences to appear. Right? So we think about DNA as mutating on a regular basis, random mutation, but at a similar rate over time. The more time 2 organisms are separated evolutionarily, right, the more opportunity there is for a mutation, because you've got more and more time. So we think logically about it, the more similar they are in DNA sequence suggests or infers that there's been the shortest period of time for them to evolve different sequences. Right, so that's the phynetic approach. So what we're doing is we're taking that matrix and turning it into a measure of difference between each of the individuals in our alignment, each of the taxa. So if you think about it, you're potentially losing information when you do that, because if we go back to our data matrix, sure, there are differences and similarities, but where those differences are and how they're distributed across our individuals is also information. Right? So if we boil those down, that data down into a measure between similarity between each of those 6 taxa, we're losing positional information and distribution information. Which ones are similar at this point as opposed to which ones are different at another point in the alignment. So one of the downsides of phonetics as an approach is you lose positional and mutationally relevant information that has evolutionary information in it. So where a change has occurred can also be used. Right? Another approach is called Phylogenetics, and that groups taxa together based on what are called shared derived characters. So basically it constructs a series of tree and tries to arrange the taxa on our tree structure so that each of the groups shares characters within the cluster. Alright, so what we should see is some consistency. This group all share a bunch of characters right from the ancestor through to that group. This group over here will share a different set of characters, but you won't have other characters that are not shared by the ancestors. So the idea being that if we've say got an A at position 233 here, all of the ancestors from that position upwards in the tree should share it. Okay, that's the basic idea. So positional and types of mutation information are taken into account in statistics. So it's using more information than phonetics. These last 2 approaches, maximum likelihood and Bayesian inference, are more statistical approaches. So basically, maximum likelihood groups the taxa together based on likelihood of fit to a specified evolutionary model. So first we construct a model of how we think that particular DNA has been evolving in terms of rates and things like that, evolutionary rates. Then we construct a series of tree and test it as a fit for the model. Take a measure of likelihood, which is a statistical approach, and then we do another tree and see whether that's more or less likely as a fit to the model. Bayesian inference is similar in I guess it's a similar concept in the way it works, but it uses estimations of probability of fit to an evolutionary model. So a slightly different form of mathematics. So we won't touch upon these really, because it's, I guess we get into phylogeny as a whole unit subject if we do that. We could teach a whole unit on just this stuff quite easily. And there are lots of people who spend a lot of time on understanding those approaches. So we think about the phonetic approaches, probably the computationally simplest. The way that works, there's 2 steps involved. First of all, we need to calculate the similarities or differences between the taxa. So most of these methods work on measures of dissimilarity. So how many differences are there between the taxa in our data set? Right. So the first thing we need is to calculate the differences. Now you would think that's pretty simple. We just count up the number of differences. But no, that's the simplest evolutionary model, right? That just by counting up what is called sequence divergence. All right? There are actually 20 or more different ways of calculating the similarities and differences between 2 sequences. And effectively they are 20 different evolutionary models. So 20 different ways in which you might get a change of base from one to another, or the insertion of a gap. Maybe changes from A to T are more likely than A to C, for example. So transitions and transversions. Transitions, so change from a purine to a purine base are often weighted differently than changes between a purine and a pyrimidine base. Right? So if we look across different organisms, we can see that the frequency and probability of different types of mutation actually are not equal. All right? Whereas if we just use a simple percentage sequence divergence, the implicit assumption is that all of those changes are equally likely, and that actually is quite unlikely. All right? So that's why there's so many different ways of calculating it, and each of them have make different assumptions about the evolutionary process. So depending on the characteristics of your data set, the similarity of the organisms you're containing, whether or not you think there's been different evolutionary rates over time between different groups of organisms that you have in your analysis, you would choose a different distance measure. Alright? So each of them, some work well under more circumstances than others, but often you'll see similar output from them. Alright? So when we do that, we're calculating measures of similarity. If we just go with the simple as to how many differences there are, I've got a little example at the top, 3 different taxa, 3 different sequences, and the differences highlighted in different colours. If we count up the differences between A and B, A and C, and B and C, and put them in a table, you can construct it like this. ABC, ABC. There's no differences between sequence A and sequence A, right? So all our all our diagonals should be 0 in that matrix. And in the lower left side, the difference between A and B is 2 bases. The difference between A and C is actually 4. And the differences between B and C is 4 as well. Alright, so that's what we call a pairwise distance matrix. So it's taking that information from our alignment, turning it into a distance matrix. This distance matrix is the first step in constructing a phonetic tree. Alright, so it's turning that information into a series of comparisons. Now this is the information that actually goes into your tree analysis, the similarities and the differences. So the second step can be a tree, or it can also be some other form of analysis. Same kind of input, but the output can be different. We can do a cluster analysis, which is a tree diagram. All right. So if we were to think of that data set, we've got differences between 2 and 4. All right. If we were to draw a tree that represents that distance matrix, it looks like that. So there are 2 units of difference between A and B. All right. So if we have branches of our tree that are all 1 unit long, you can see it's 1, 2 between A and B. Difference between A and C is 1, 1 plus 2. That's 4. All right. So again, this is something you can construct purely from logic, but it gets rapidly complicated. The more taxa, the more things you're comparing, it's really difficult to build trees manually like that. So again, there are computing algorithms that will generate the best fit tree structure or cladogram, cluster analysis, to fit the input data, which is the distance matrix. So in goes the distance matrix, out comes a tree structure. So there's a couple of different examples. Again, there's probably 10 or 15 different ways that you could take a distance matrix and create a cluster diagram. All right, and again, when you do this kind of analysis, you need to choose an appropriate clustering algorithm depending on the nature of your data set. So 2 common ways, what's called unweighted pair group mean average. So that's an algorithm that pairs up the 2 closest, averages them, and then compares them to the next closest. It builds a tree successively from the closest to the most distance. There's another method which is a little more sophisticated, called neighbour joining, probably the most common tree building approach used in phonetics. So this kind of method. So we can take our distance matrix and we can turn it into a tree structure up there, or if we think about this, we can also do it in 2-dimensional space. All right, so that's what's called an ordination analysis. Same input information, instead of arranging them in a network diagram, a tree, we can arrange them in space. All right, so we've got 2 axes, and we can arrange dots in that space so that the distance between A and B is 2, the distance between A and C is 4, and the distance between B and C is 4. All right, so just be aware there's different ways of looking at data. Trees are great for if you're really interested in evolutionary relationships, but sometimes You're interested in similarities and difference where the relationship between organisms may not reflect that evolution by descent process. And a good example is population genetics, which is what we'll cover in the next lecture. We're going to compare genetic similarities and difference between individuals that are part of the same species, for example. The relationships between those individuals is not evolutionary, it's population, right? So they're recombining genetic information all the time. Right? So this is not really a very useful way of looking at relationships among individuals within a population, because it's not a process that can be reduced to a tree-like concept, whereas individuals that are very different can be further away in two-dimensional space, for example. And logically, when you look at that diagram, if we've got three all clustered here and one over here somewhere, you can still visualise that these ones are more similar and this one's more different without needing to confine it to this tree-like concept. So just be aware that the same input can be visualised in different ways for different purposes. One of the benefits of these phonetic approaches is they're computationally a lot less intensive. So if we've got a big data set, thousands of species, thousands of base pairs of DNA, this is probably the only way that you can do it without a bit of supercomputing, because it can take days or months to run some of the other more intensive approaches, whereas phonetic approaches, it might take several minutes perhaps to run a huge data set, or a few seconds for a data set the size that we'll be working with this afternoon. So it's basically an iterative mathematical process that builds the tree in an ordered way. The downside, phonetics make some assumptions about the evolutionary process that are rarely valid, depending on what kind of clustering method and distance similarity method you use. The other downside of phonetic approaches is that loss of information at the start. So you lose positional and mutation information from your alignment as soon as you boil it down to a single comparison between each sequence, which is what all phonetic approaches do. So just be aware that there are a couple of downsides, but under normal circumstances between organisms that are vastly different and may not have vastly different evolutionary rates over their evolutionary history, a phonetic type approach will actually recover a very similar tree to some of the more sophisticated methods, and you can do it much more quickly because it's computationally a lot easier. So for that reason, quite a lot of analyses you might start by running a phonetic method to build a tree, and then gradually use more sophisticated methods and look for similarities and differences between the methods. In fact, some of those other methods, like maximum likelihood, actually build what's called a guide tree first. So they actually build up a tree using a phonetic approach and then use that as a starting point and modify the tree through a series of processes. Right? And that's one way of making those more sophisticated algorithms a little bit faster. Okay, so different tree building methods, just to show you that they don't end up with the same result. So here I've run the same input data set through, so it's the same distance matrix through a UPGMA cluster or a neighbour joining cluster. You can identify actually similar groupings. So we've got a grouping of loach, which is a fish, carp, frog and chicken. So we've got loach, carp, frog and chicken up there. All right, down the bottom here we've got mouse, rat, seal, cow and whale. Mouse, rat, seal, cow and whale. So those two major groupings still exist in that tree. So some elements of the tree are very similar. However, in this tree, humans sort of pop out somewhere in the middle, as opposed to on the outside there, but you can see that the general structure is not too dissimilar, but the way they're arranged is a bit different. The other thing about UPGMA, it makes an implicit assumption about evolutionary rates, which means you always end up with this kind of flat branch effect. Alright, so basically UPGMA as a clustering algorithm assumes a constant evolutionary rate across all the groups. These groups are a pretty broad group of vertebrates that have been around for 500 million years. The likelihood of the evolutionary rate being consistent and constant across such a diverse group of organisms is actually 0. Alright, so UPGMA probably doesn't actually fit that assumption well, but you can see it still recovers some of the major clusters, just in a different way to neighbour joining. That's an example of how different assumptions are implicit in the way you analyse the data. I just want to finish off talking about what's called rooting your tree. Alright, so we look outside at a tree, it's quite clear where the root is. It's down at the bottom. Alright? But what we're constructing when we do a clustering algorithm is just a mathematical network. A set of numbers that describe the lengths of these branches and how they're connected to each other. There is no up, down, past, or now in the networks that we're constructing. We have to actually reorient that network in some way. And there are a couple of different ways of doing it. If we want to interpret it as the bottom being early in time, in the past, and the top being now, the present, when we've taken our individuals and sampled them. Alright, so that's what we call placing the root. So that's what provides the time and orientation to the tree. Alright, so effectively when we root the network, we're basically taking a point on the tree, saying, well, that's the bottom. Let's bend all those branches up away from that point. There are a couple of ways we can do this. One of the simplest is what's called the midpoint root. And this basically concept is that the middle of the longest path through the network must be somewhere near the bottom. If you think about the logic of that, you can kind of see why that might work. Alright? So if we were to draw the longest path through this tree, it could be from human through to chicken perhaps, or more likely probably from something like fish through to humans. We know that they're probably the most divergent from our fossil history. So somewhere in here is the midpoint root. We take that point, bend all of those up away from that point, then effectively we're rooting the tree at this point. Right? That midpoint idea is only true if the evolutionary rate is equal across your whole data set. It won't work through the logic, but if you think about it, that's effectively what it's doing. Right? And that's rarely valid, right? Unless your organisms are really closely related and they haven't been through any, I guess, strong selective forces that act differently on some groups than others. All right? A better option, rather than midpoint, and you'd really only use the midpoint approach if you really had no other means or information to choose. If you had nothing else to go on, you might use a midpoint root. A better option is to use some other form of information. So when I was talking about, gosh, where would the longest path through this tree be? I thought, well, logically, fishes evolved considerably earlier than humans. So they're probably the most divergent groups. So I'm actually using external information. It's not part of my data set. It hasn't been used to generate this network, right, which has been based on some gene sequences. So I've taken some external information, the fossil record, to say, well, where should the root be? Right, that's a far safer way. If you have reliable other information that you can use to choose the position of the bottom of your network, is always the best way to go. And you can do that using things like fossil records, but you don't always have a complete or any fossil record, depending on the organism you're working with. So another way we could do that, if we don't have fossil record to work on, is to use what's called outgroup routing. Alright? So routing using an outgroup basically needs you to determine what an outgroup and an in-group are. Alright? So if we think about outgroup and in-group actually is the organisms that we're interested in. So this afternoon, we're interested in the relationship between our Vibrio isolates. Right? So we can think about the genus Vibrio as being our in-group. Right? So all the Vibrio that are in our data set, they're part of the in-group. If we want to determine where the base or the root of that network might be, then we can include an outgroup, something that is not part of the group we're interested in. Right? So the definition of an outgroup is a comparison taxon or organism that we definitely know is not part of our in-group. So Vibrio, it's part of the gamma proteobacteria. A good choice would be something that's not a Vibrio, right? Because then there's no chance it's part of the group we're actually examining in our analysis, all right? So you can choose your outgroup based on the nature of the group you're interested in. So if we're interested in Vibrio, we could choose another gamma proteobacteria, and if you know your bacterial taxonomy at all, we could probably use something like an ultramonid species because we know they're relatively closely related to Vibrio. They're definitely not a Vibrio. And I'll tell you why in a minute, why that's important. We then include that in our data set, so we put it in our alignment, we calculate the similarities and distances and build our tree with our outgroup in our network. Then we can use that to orient our tree. So here's an example, same data set from the previous slide, but I've changed the outgroup. All right, so I've taken the same data set and I've told the analysis this is our outgroup, and then in the other analysis I said no, use this as the outgroup. There the outgroup was frogs, here it was whales and cows. Now those networks are identical, but all that's happened is we've bent the branches around from a different point. From this point here, whales and cows, and from that point up there, using frog as the bottom of the tree. Right? You can still see some of the same groupings in there, but the way you would interpret that analysis is quite different depending on the way you, which organism you use as the base of the tree. Right? So actually, choosing a root for your tree is a crucial part of getting something that makes sense and is interpretable. So if we change that outgroup, we get very different impressions of the evolutionary relationships. Okay, so what's the concept behind using an outgroup? Here's my go at trying to explain it. So here's our group, which could be our Vibrio, right? A little cluster of our organisms that we're comparing. Logically, they've evolved somewhere back down here from a common ancestor of this little cluster of organisms we're interested in. If we take a group of organisms that are closely related, but definitely not part of that group, logically they're closely related, so somewhere back down here, they also had a common ancestor with our group of interest, okay? So somewhere down here is the root of the tree. All right, so where this group joins into that network, then we can assume must be relatively close to the true root of the tree. All right, do you understand the logic there? But let's say we choose an even more distant outgroup for rooting our tree. All right, we use this group here instead of this one in the middle. Well, we can still estimate the root, but we've got a much longer period of time and therefore a much higher level of uncertainty as to where this joins that and whether it is an accurate placement. All right? So you've got all that evolutionary time and distance that you can't necessarily be sure you've been able to estimate using that network. So logically, your outgroup doesn't want to be part of this group, because if it is, it will reorient the tree almost upside down, if you think about it. If we take an organism in here and use that as the bottom, all the tree gets bent up in reverse, all right? So it needs to be outside our in-group, and ideally as close as possible to this, but definitely not part of the group we're interested in, all right? Otherwise, things get messy. So we don't want a really distant group. And in fact, the way most people would do outgroup analysis is to include a series of organisms of increasing genetic distance that they were sure were not part of the in-group, to provide some structure down here, to provide higher confidence that we could estimate where this point was, right, which is the base of our group of interest. Right, so that's the logic behind using outgroups. So what I've been showing you are what are called cladograms. So little structures that just indicate the level of relationship. Closest, next closest, next closest, where the length of those different parts of the network are not varying. They're just levels, 1, 2, 3 levels of relationship. Those branch lengths in a cladogram generally don't have any meaning except level of cluster, either closest or next closest. Most of the time what we want to produce is what's called a phylogram. Right? So both the order at which things join the network is important, but also the length of the branches. So you can see in this tree, some branches are shorter, some are longer. So there we can draw networks that actually reflect the genetic difference. So if we were to look at the difference between eukaryote 1 and 2, it's the sum of that length, that length, that length, and that length. And ideally, our tree, using a phonetic approach, all those lengths should match or be scaled to the same as the input matrix. Right? So what we're trying to do is reflect the distance matrix in a tree-like structure. So I just want to finish with some caution. So phylogenetic analysis, when you first start, tends to be very much a black box exercise. You process the data, you put it in, and out comes the tree. And this is actually even stated in a quite classic text on molecular systematics. Phylogenetic analysis is treated as a black box into which data, which has often cost a lot of money to collect, fed, and out of which the tree springs. You shouldn't consider phylogenetic analysis a black box. Right? You actually have control of how that black box works, and you should be looking to at least test the stability and reproducibility of the analyses using different approaches. If not, try and optimise the evolutionary model to be a good fit to the data set and the animals you're working with. I'll finish with an example, which is ribosomal RNA, which is what we're working with. We will do a standard phonetic analytical approach, but just be aware this molecule is really complicated. So it's not a coding molecule for a protein. Here's a secondary structure of a ribosomal RNA. This is our strand of RNA, and it forms stem and loop regions. And these stem regions are held together by the AU-GC pairing, right? So the pyrimine and the pyrimidine bases. And there's loop regions like this, which are not held together by paired joins. So if we look at this whole molecule, there are regions that are conserved, so there's very little variation, and they're the bits that are actually functionally really important, otherwise the molecule doesn't do its job. Remember, it's part of the ribosome. There are regions that vary, and there's also what are called hypervariable regions, these blue regions, that are not so important to the function of the molecule, so you get a lot of mutations in those regions. So a simple evolutionary model where every mutation, position and type is just as likely clearly isn't a good solution for this molecule. Right? There's even compensatory changes in the stem regions, right? Because these have to pair up to hold the molecule together. You tend to have mutations, say from a G to an A, will be reflected by a C to a T on the other side of the secondary structure where it needs to match up. Right? So if we were to use an evolutionary model that reflected what we understand to be the process of mutation and evolution of these molecules, we would need a model that would take into account all of these variabilities. And that's a hideously complex model, particularly these ones, compensatory changes. Because, for instance, this stem here is probably 30 or 40 base pairs away from the compensatory change on the molecule. So up here we get a change from a G to a T. Over here, 20, 30 base pairs away, the compensatory change happens. So rather than being independent mutations, they're actually linked in some way. Right? So, short answer, even this very commonly studied molecule to do phylogeny actually should really use a really complex model for a thorough and robust analysis. Having said that, you can actually get pretty good approximations by using simpler models, and that's actually what most people do. Very few studies that actually incorporate all of these elements in the way they analyse the molecule.